My name's Steve Hunter. I'm the product manager for preclinical and optical microscope imaging systems. And today we're going to do a presentation on the Longitudinal next generation photoacoustic system. My co-panelists for this are Dr. Yasufumi Asao and Mr. Yoshiharu Takahashi. I'm going to give the presentation, but they'll be there at the end to answer any questions you may have. Please put forward your questions if you think of them during the presentation, or wait to the end and we'll open it up to people asking. Anyway, I will share my presentation now and we'll get underway. Right. So today's webinar, as I said, is about the Longitudinal next generation photoacoustic system. We've had this product with us for a couple of years, and I've been very impressed with the quality of the images that it produces. And I think it is a differentiator from the other products in this area in the market. So today I'm going to give an overview about the technology, some examples that have been done on imaging with this system, and then some concluding remarks. So we'll start with the overview of the system. As it stands at the moment, the system also has a sister product, which is for clinical imaging. In fact, that product has been licensed to be used as a medical imaging device in the Japanese domestic market. So it is capable of imaging quite a wide range of areas and tissues. And you can see here in the diagram that they're showing human limbs, calf limb, human palm, but also small animals from the rat down to small nude mice. What we're going to concern ourselves today is with the preclinical system. Most of the data that's going to be shown is based upon images generated from the signal from hemoglobin in the red blood cells in the various animals. The system itself includes a hemispherical detector array, which scans underneath the tissue, in this case a hand, and below that is a laser which provides the stimulation to produce the ultrasound signal, and a small camera to actually take an image outline of the tissue in question. Now, when this hemispherical data array, as you can see here below, you can see that there's a small orifice in the center for the laser, and then next to it, the orifice for the camera. And the array itself has 512 microphones, detectors, in a fixed pattern, which don't shift from that orientation. When the laser excites, in this case, the red blood cells in the blood vessels, it produces a combined wavefront. If the blood vessel is in the horizontal position, we get predominantly a signal, as you can see here, across the center of the detector array. But should the blood vessel be at other oblique angles, then this predominant wavefront moves further and further towards the periphery. This, along with signal processing, allows us to produce a rather anatomically correct image, as you can see here. In this slide here, we can see that if we have a spherical ring object being excited, then the 3D image that's produced is in fact a circle. If we compare this to lineal arrays, which is pretty much in every other photoacoustic imaging system in the market, the cross-sectional image is a sort of distorted version of this circle. Even though it provides imaging information, it is in fact not an anatomically correct image. As I said earlier, the first system from which the preclinical has been derived was for clinical research, and currently there's been already some studies done on this in three major areas. One looking, which say planning for free flap, which is actually for looking at the quality and viability of tissue for tissue generation, breast cancer investigations, and also investigations of lymphatic systems. So these are the first early clinical applications. For preclinical applications, this is still fairly early days, but you'll see in some of the imaging examples later that this does show a lot of promise. Now, the medical system, although it's conceptually identical, is built into a elongated horizontal bed type system, which for obvious reasons is how we'd want it for imaging people. But the preclinical system is built into a sort of two-story structure, which is much more convenient for the research lab. And in fact, it's got quite a small footprint. As you can see, this is a system here. It has a width of 1.6 meters, a depth of 1.1 meters, and a height of 1.1 meters, making it very convenient to stand and conduct experiments with. This is showing an actual animal being scanned. And the things I want to point out in this slide are that you'll notice that there's actually a small reservoir of water on that the animal sits in quite comfortably, and that the hemispherical detector array scans underneath the animal and moves around to conduct quite a large area. In fact, even though the nominal field of view of the detector array is 22 by 22 millimeters, the actual largest scan area that we can scan with the system is 290 millimeters by 180 millimeters. A scan of that size would take about 4.5 minutes approximately to scan. But if we want to look at what this system is capable of in terms of its temporal resolution, then if we kept the detector array static, we can actually do a maximum of 30 frames per second with the system. And remember that these are actually three-dimensional images in a 22 by 22 millimeter size. And to further emphasize the anatomically very accurate images that the system provides, this is an example where we're looking at published images from Ithera, one of its competitors, and Tumor, another, showing these representative images that they actually use for marketing purposes, compared to the sort of image that we get from the Luxanos system. And also further emphasizing this anatomically correct imaging, we can see that the camera picks up an outline, and then we superimpose the arterial and venous systems over that, onto that image, and compare it probably to the sort of images we get from a dissection, or in fact, it's probably more closely related to the outline and imaging that we'd get from an IVUS system. But it's really very good in terms of producing undistorted images of the sample. Now we'll go through a few imaging examples with the system. Now these are done with post-mortem animals. So these are a few minutes after the animal's been culled. And even though the animal is dead, there's still enough signal from the arterial and venous systems to produce a quite distinct image. We can see that there's a lot of detail in these images. This is the image of the lower face of a nude mouse. And likewise, this is looking at a rat heart. And what is quite surprising is that the sensitivity of this system is even able to image the cerebral blood vessels of a hairless rat through its skull bone. So again, quite remarkable. The other advantage of the system, because of the large scan areas available to us, is that we can image a large. Don't have to do just a single mouse. We can actually do multiple animals. This was just a trial run done with a single nose cone, rat nose cone. But as you can see, that we can get well-defined images from 2 mice. So we don't know exactly how many animals we could do, but possibly 4 or 5 in a certainly mice in a single session. So to look at the performance of the system as a 3D imaging device, what's been done in this image, this has been done with a single wavelength, not looking at the saturation, but just the signal from the blood vessels, is to superimpose a lookup table showing the depth into the tissue. And in fact, we go to the next image, we can see that we look through a cross section here, we're imaging down to about 10 millimeters. So we feel the system's capable of even a greater imaging depth, but this will certainly be more than adequate for any mouse experiments. So the blue is actually that nearest the laser, and the red, the deepest part into the tissue. In terms of spatial resolution, we're quite comfortable in choosing about 100 microns resolution in imaging. And this was confirmed from this image of the liver, looking at, after dissection, the same blood vessels, and noting that they were measured after dissection at 0.1 millimeter. Now, the current derivation of the instrument has 2 lasers. They're at 797 nanometers and 756 nanometers. And if you look at this graph showing the relative absorptions of deoxyhemoglobin and oxyhemoglobin, these wavelengths are selected to look at a large difference between the absorption of the laser at 756 and then no difference in the absorption at 797 nanometers. This is then plugged into an expression looking at these differential absorptions to produce what's called an S factor or saturation factor, which is looking at the oxygen saturation of the blood based upon the relative absorptions of the various lasers in producing a photoacoustic signal at these 2 wavelengths. Now, this is a relative, not an absolute figure, but even so, it produces very consistent results. So here we're looking at the abdomen of a rat, and looking at the a mouse, 9-week-old female. And then if we look at this, we can actually pinpoint out a vein and an artery, and looking at the relative saturations at those 2 points. And again, another mouse in the dorsal position. And here we're looking at, I think, the kidney when it gets to that. Sorry, it's a little bit of a long animation. And here showing two veins and an artery in the kidney of the mouse. The S factor is very useful in being able to determine various anatomical features. If we look at it with no S factor, we can certainly make out various anatomical features, but this is a lot clearer once we apply the S factor. And then veins and arteries are very easy to see, and it also makes the being able to pinpoint liver and spleen, etc., a bit easier. Interestingly, the S factor is useful after looking at euthanized animals. And these are two animals where we're looking at the alive view and the dead view in both instances. As you can see, there's a lot more detail in the live mouse, but still some useful information being able to be produced from the dead animals, which are done about approximately 5 to 10 minutes after being culled. Now we'll look at some examples of tumor imaging. The first two examples are actually from both euthanized animals about 5 minutes after being culled. And here we're looking at a sarcoma L-line, which is xenographed to the hind limb of the animal, showing some quite nice detail of the tumor. The next two are breast cancer cell lines, which have been in the first instance on the mammary gland of the animal. And what's interesting to note with this one, even though it is a euthanized animal, we can see that there's a large dark area here, which is showing most likely necrotic, or at least tissue that is got very little oxygenation in it. The third example is actually in a live mouse. And here we can see some really good images of the angiogenesis in the tumor. This was the same breast cancer cell line, but done on the back of the animal. Another example of tumor imaging, again, was using the 4T1 breast cell cancer cell line. And in this case, xenographed into the mammary glands on the right and left side of the animal, a bulb C mouse. And what we've done is taken a region of interest here, and then looked through that region of interest on the right and the left. And we can see that there is significant hypoxia within the center of both these tumors by this investigation. The other good feature of this system, because it is very, I'd say, supportive of longitudinal studies, because it's very easy and very gentle on the animals in terms of the way we do the imaging, it's possible to do long studies with animals. In this instance, we've done a study over 11 days of 3 different animals. And I should point out that day 6 is missing because it was actually a day that was a public holiday. Now, looking at this data, we've looked at two things. One is looking at the saturation of veins and arteries in the liver of all three animals. And you can see that there's a pretty consistent S factor, so saturation factor, from both the veins and the arteries over a 11-day period of time. But probably of more interest is looking at the saturation factor when a cross-sectional region of interest is performed through the tumor. And in this case, this is one example where we can again see this hypoxic, if not possibly necrotic area in here, and that this change occurred at almost in all three instances at about 9 days, and it was quite marked drop in the saturation factor, showing a significant change in the tumor growth patterns through these from day 9 to day 11. So in concluding, I want to point out that this is still a relatively young technology. The first iteration with a single wavelength was only released in 2021, and it was 2022 when the fixed 2 wavelengths system was released. The next iteration of the system, due for, I think, release fairly soon, will have a tunable laser, which is going to make the system more capable in terms of doing molecular imaging, whereas we can add other photoacoustic contrast agents and tune the laser for those agents, allowing us to do a lot more studies in terms of targeting different organs and molecules within the animals. So in summarizing the advantages of the system, it's very safe. The animals are not really sort of disturbed in any way other than the anesthesia, and very safe for the operators consequently, and compared to doing things like PET or SPECT imaging. It requires very little in terms of infrastructure costs, just basically some floor space. Experiments are easy to set up. We can do quite a high throughput. We can scan quite a large area, and it is really ideal for longitudinal studies. In terms of animal welfare and ethics, again, there's very little stress on the animals, so ethics should not be an issue with this system. So finally, I want to thank you all for your attention and attendance today, particularly from the team at Luxanos and from the team here at AXT. So thank you all. And I'll stop sharing the screen and open it up for questions. Are there any questions? Just a moment, I'll invite in my co-host, and they might be better at answering this question. The question was, what would the S factor compare to what the Vivo laser system uses for producing oxygenation numbers?
Let's go on with it. Hello. So I'm afraid so we don't know much about the Vivo systems the oxygen numbers. So we're gonna check it and get back to you. Is it okay?
Is that all right?
So we don't know much about oxygen number of level laser. So we're gonna check it and get back to you.
I've opened everyone up to talking now, so if you want to chip in and ask questions as well. The second question was, may I know any clinical applications in humans at this moment? From Walter Liu.
Yes, like you explained, so we are working for the mainly working for the clinical application, and those are for plastic surgery and reconstructive surgery for breast, and also the lymphedema surgery or those kind of things.
Yeah. And the third question is from Josh Bourne. He asked, people use ultrasound to damage the blood-brain barrier. Is there any evidence that vessel integrity is damaged during imaging? I could probably chip in a little bit here. The answer is probably no, because in that type of work, and we actually do sell that type of technology with our MRIs, there's actually a highly focused fairly intense ultrasound system to do that. In this case, we're not introducing ultrasound. What we're doing is introducing a laser, which actually then, through a sort of a thermal effect, generates an ultrasound wave that we are in fact detecting. Asaya-san, would you like to possibly comment as well on that?
Respiration rate.
Is there any damage done to?
うん、このイメージの、外部イメージの、まあ多少、
影響しますね。だから大盛りを少し乗せてたら、その下は安定するんですけど、やっぱりちょっと
あとはソフトで直すということですかね。
ちょっと今はさせてほしいです。
Yes, actually, so respiration little bit affect to the image quality. So therefore, so we use some weight to push to give the pressure on the mouth. It's not too heavy, but to stabilize the image.
Does that answer your question, Nick? Are there any more questions, Nick? Thanks. Great. Thanks, Nick. Does anyone else have any questions at this point?
違うレーザーです。メンテナンスは入れます。
Can I ask one question, Takahashi-san?
Okay.
When do you plan to release the next system with the tunable laser?
Yes, tunable laser, it's we got a contract with the laser manufacturer. So if you prefer, so we can manufacture a tunable laser maybe in 6 months of time.
Oh, great. That's good to know. Another question is, do the lasers in the system require any kind of maintenance? Are these flash lamp based lasers?
Yes, it necessary to tune the laser at least once a year, like once a year.
Right. But there's no ongoing maintenance of the lasers otherwise. there's no cooling systems or anything else of this nature.
Oh, of course, to 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 use the machine in the best condition, not only the laser, other mechanical parts and electrical parts should be checked at least once a year, so somewhere.
Okay. So do we have any more questions? Looks like not. So again, I want to thank everyone for attending, and I hope it was informative. And if you have any further questions, please contact me at AXT, and I'll be more than happy to get you those answers. So thank you very much. And at this point, I will finish the webinar. Thank you, everyone.